fatigue cohort. This is a cohort of young adolescents that were infected with Epstein-Barr virus, getting infectious mononucleosis, some recovering and some not. So this is also a very interesting presentation that really is trying to get at some predictive markers for CFS. We'll end our program with Dikoma Shanggu from Weill Medical College of Cornell University in Manhattan. Dikoma has been a funded investigator of the CFIDS Association for a number of years now, and he has been using a very powerful imaging technique to try to identify biomarkers in CFS. And again, he has developed over the years a very fascinating model of CFS pathogenesis, and we're excited to bring all of these speakers together because I think by the end you're going to see some common threads through these sets of talks. So today's format will be Kathy and Alan presenting first, then Gordon, then Dikoma, and we'll wrap up with hopefully some time for a few questions or discussion amongst all of us. So without further ado, I'd like to introduce Kathy and Alan.
Hello. Hello. Thank you so much for inviting us to do this, and I have to begin by saying once again, as I have said every time I get a chance, how grateful we are to CFIDS for providing us this very, very critical support for our research. Our project was called Novel Ion Channel-Based Biomarkers in Chronic Fatigue Syndrome. I'm Kathy, I go by. This is my husband, Alan, and I. This is the first time in well over 30 years of marriage that we found a project important enough that we were willing to collaborate together on it. So this is a very big deal to both of us. We also have probably what I consider one of the very, very best clinicians in the chronic fatigue area to work with us, Lucinda Bateman. We have an exercise scientist, also very knowledgeable, Andrea White, and Ron Hugan is our lab specialist. And all of this work is being done at this present time with CFIDS support. We did begin our work with an R21 grant from NIH, but it would have stopped dead in its tracks if we hadn't then received the CFIDS grant when we did. I do want to note that the findings that I'm presenting here, because we were collecting data right up and through last week, we will be adding more data before the final analyses are done. So the findings are somewhat preliminary, although these were just the last few subjects. I also want to acknowledge a potential conflict of interest because we do have a patent pending, and I'm just going to say that this is actually a very hopeful thing because what the patent has to do with is using these biomarkers potentially for clinical applications in chronic fatigue syndrome. So I acknowledge that, and at the same time, it's hopeful, not anything negative. Okay, what we had done before, quite separately, Alan and myself, was to look at muscle fatigue and pain in quite different ways. Alan had always used animal models, and in his animal research, he had found that molecular ion channel receptors were very important to detect muscle fatigue and pain, but they had to be 2 or more of them working together and activated together to do this. These particular receptors detect acid, lactate, and ATP that are produced by muscle activity, but they're produced even in very, very low level activity like sitting, and then more and more and more produced as the muscles become more and more activated. And these particular receptors are known by these little acronyms, ASIC standing for acid sensing ion channel, P2X, those are purinergic receptors that respond to ATP, and the TRPV1 is actually called the capsaicin receptor, and it responds we think to heat as a byproduct of activity, but maybe to many other things as well. Now, in my research, I've always done human research with healthy individuals and with patients. And my research had looked at the importance of the sympathetic or adrenergic nervous system in terms of its role in fibromyalgia pain. And we had done a study where we particularly tried to block that pain by giving an adrenergic blocking drug. And we were able to show that there was a little dose of this adrenergic drug reduced pain. So that's the background. So we moved from that to our study of chronic fatigue syndrome, and we began with the central theme that mental and physical fatigue and muscle pain increase and often persist for 48 hours or more after exercise if you have chronic fatigue syndrome. And the exercise doesn't have to be vigorous. It can be fairly mild. So we wanted to know whether gene expression changes that we could monitor on the white blood cells in blood samples, whether we could look at changes in specific receptors, and we focused on those same ion channel receptors that were important in the animal research and the adrenergic receptors that were important in the fibromyalgia research. And we also, because it wasn't difficult to do, we added in some immune markers because those have also been identified in other research involving chronic fatigue syndrome. So we looked at the responses following exercise at a very mild level using this exact exercise bike, which makes whole body exercise, arms and legs moving together. And the first set of our results were reported in the Journal of Pain last year, but I'm going to talk about what we found since then. Are we going to? There we go. Okay, so you're going to see a very complicated slide here. I apologize for its complications, but we made it very colorful and pretty. So what you're going to see, at the top here are the responses to 25 minutes of exercise in healthy control people. And they were exercised at a moderate level, that is 70% of their predicted maximum heart rate. And you'll see we have baseline, everything is corrected to what each person's individual baseline was. Then their responses when we sampled at 30 minutes after exercise, 8 hours after exercise, 24 hours after exercise, and 48 hours after exercise. And the blue-colored ones are those sensory ion channel receptors. The kind of red-toned ones are the sympathetic or adrenergic-related genes. And the green-toned ones are the immune genes. But the thing to point out here is that aside from a little tiny blip in the beta-1 adrenergic receptor at 8 hours, not much is going on in these healthy controls. Moderate exercise doesn't do much to them. Now, the next thing you're going to see are chronic fatigue patients. In this case, we're focusing on a specific subgroup, those who have comorbid fibromyalgia. And they're exercised at the same level, 70% of their predicted maximum heart rate. The first thing that you're going to see here are those blue-toned ones, the ion channel receptors coming up. And you can see there are dramatic increases beginning right away at 30 minutes after the task is done and lasting through 48 hours. And the biggest effect is in those purinergic P2X4 receptors, that's very kind of bright greeny blue one. Then the next thing you're going to see are the adrenergic receptors. Very, very large scale increases in gene expression of those adrenergic receptors, both alpha and beta. And the big one there is the beta-1, but actually for statistical significance, beta-2 is just as big. And then there are more modest increases in the immune markers, IL-6, IL-10, and so forth. Now, we also compared these to individuals who had chronic fatigue syndrome without comorbid fibromyalgia. And I hope that's coming up for you. What you're going to see is that it really is a remarkably similar pattern overall. And there's only a few genes that seem to differentiate these two subtypes of chronic fatigue syndrome. And those are going to be indicated by arrows coming down at all the different time points. And the ones that are different are the ASIC3, the very first gene that you see there. And what happens is ASIC3 is increased if you have comorbid fibromyalgia, but not if you do not. And the TRPV1 or capsaicin receptor also increased if you have comorbid fibromyalgia and not if you do not. However, all of the other genes, including that big P2X4 increase and the adrenergic increases, all are common across both of the subgroups. So we also wanted to compare to another patient group that experiences a great deal of fatigue, because we wanted to be sure that this profile was very good at discriminating specifically chronic fatigue syndrome. So we recruited a group of multiple sclerosis patients, and these MS patients showed no changes in those sensory ion channel receptors, those blue-toned ones. They showed a very brief transitory kind of increase in beta-1 receptors at 8 hours, but nothing like the prolonged and large-scale adrenergic activation that we saw in the chronic fatigue patients. Then we also recruited a group of healthy controls who were exercised at a much more strenuous level than the patients had been. And this was in order to correct for the possibility that we simply were physically challenging chronic fatigue patients way more than we were challenging our healthy controls. So exercising healthy controls at 85% of their predicted maximum heart rate still did not produce this large-scale activation in these genes. The only thing we saw there is, again, this little spike of the beta-1 receptors, very brief and transitory at 8 hours, nothing else similar. Okay, the next issue that we wanted to address was whether we could use our profile of gene expression changes to determine whether a medication might be causing beneficial effects. And so what we did in this, with the help of the CFIDS support, we had previously only studied individuals who had been off all of their medications, all their pain medications. And what we did with the CFIDS support is we recruited some individuals who were tested while they were on their currently FDA-approved pain medications. And what we found is, even though there were just a few of them, and these results, therefore, are really, really preliminary, but we did see that at the very least, we were seeing beneficial changes. That if you look on the left, there is much less of an increase in those red-toned adrenergic receptors. If you look on the right, where we have an age, gender, and severity-matched group who were not on this FDA-approved pain medication, they have much bigger adrenergic increases. It didn't seem to have quite the same huge effects on the sensory ion channel receptors, but there were some subtle changes. And we think we're going to extend this work and hopefully see something more dramatic. The other issue that we wanted to address was whether our profile would reflect clinical severity or disability of CFS. And with our clinician, Dr. Bateman, she blindly rated the level of disability and clinical severity of these patients. They were all her patients, so she had really, really extensive medical files on them, and she divided them into 4 groups. And what you see on this slide is that the most severely affected group, the one in red on your left, did indeed show overall the largest average increases in, this is just summed across all the genes in our profile. And group 2, which was the next most severely affected, the gold group, had the next greatest set of increases. And then when we got out to group 3 and group 4, who had less severe severe symptoms and less severe disability, they still showed significant increases. I don't want to imply they did not, but it does show that their genes increased less after this exercise test, so that we did seem to be marking the level of disability as well. So to sum this up, the support that we got from CFIDS made all of the following possible. We were able to demonstrate that our gene expression biomarkers had significant utility for distinguishing chronic fatigue patients who had chronic fatigue alone from healthy subjects and also from those with MS. And we also were able to distinguish those people from individuals who had both chronic fatigue syndrome and fibromyalgia. So all of those groups had different profiles. We also generated preliminary evidence that our gene expression profile is related to the severity of the disorder, and that it can be used possibly in studies to look at the effectiveness of treatment as objective markers along with the more subjective ratings of improvements of symptoms. Now, some of the things that I want to emphasize that I couldn't show in our other slides is that the CFIDS support helped us begin a very important collaboration to move into a different area, but one that's very important to chronic fatigue syndrome. We are now collaborating with another University of Utah expert in murine retroviruses, Dr. Ila Singh, and we are working with her to look at those retroviruses in our chronic fatigue syndrome population. We also have received a grant from Pfizer to continue to study our gene expression profile after exercise as a potential objective marker of their well-marketed treatment for chronic fatigue syndrome plus fibromyalgia. It's actually designed as a treatment for fibromyalgia, but the patients who have chronic fatigue syndrome plus fibromyalgia are an important population for them to study. And the other thing that we did, we've gotten to the point where we're so excited about our findings, but it's hard to know whether to write papers first or submit grants first. But this year we worked on submitting grants. We submitted 5 separate grants to NIH to expand our research on this. We wanted to add some additional genes to our biomarker profile, especially ones that, and also to include retrovirus infection-related markers. We also have basic animal research that we want to keep funding to go on looking at the mechanisms of sensory fatigue, and we're trying to make sure that our profile is truly specific for chronic fatigue syndrome. So we want to compare to patients with clinical depression, to fatigue of aging, to cancer-related fatigue, and to fatigue related to heart failure. All of that, someday we hope we will get funded. And once again, thank you so much, CFIDS. Thank you, Suzanne. Thank you, Kim.
And thank you, all of our participants, all of our listeners, all of our supporters of the CFIDS Association who have really made this kind of research possible. If it weren't for you, we wouldn't be able to support this kind of phenomenal research. So I've jotted down a couple of questions, but I'm going to save them to the end. So let's move on to Dr. Gordon Broderick, who's going to tell us some of his research findings on immune markers in post-infectious fatigue syndrome. Gordon, are you there? And Kathy and Alan, if you could put your phone on mute or your mic on mute. Gordon, are you still there?
Yes, I'm here.
There you go. Oh, there he is.
I'm just collapsing my various control panels here.
Was that wonderful picture taken in Canada, or is that in Chicago?
It was taken in Banff.
Oh, that is absolutely handsome. Very nice.
In the Canadian Rockies. Well, it was getting a little chilly, so it was a brief shoot.
Very good. Well, without further ado, Gordon, take it away.
Well, thank you again for this kind of invitation to speak. I just want to emphasize that we are still very active in this project. We have applied for an extension without additional funding and been graciously granted that extension. And we are still active on several fronts in this project, but I will present some of our preliminary findings. When we were first awarded this grant, we immediately started ramping up our efforts in developing the experimental protocols and computational tools that we knew we would need to analyze these samples. And to do that, we entered into a collaboration with another CFIDS-sponsored team, the team of Nancy Klimas and Marianne Fletcher, and used their cohort of adult female CFS patients as a testing ground and a development ground for some of the numerical tools that we are now applying to the post-infectious fatigue cohort. So one of the first things that we did, one of the first data sets that we looked at was the expression of a broad panel of immune proteins. And we were less interested in the expression of individual immune proteins than we were in how these immune proteins express in relation to one another. So what are the patterns with which they co-express? And we analyzed this by constructing association networks that essentially map out this coordinated expression of cytokines. What we have here on the left-hand side of the slide are two cytokine association networks, or immune protein association networks, one for the healthy control cohort, one for the chronically fatigued. These are adult female patients. And just visually, I think it's striking at how the geography is different. Right away, we see the emergence of a very tightly connected module of cytokines that are composed or essentially represent the balance between a viral and allergic response. This module is tethered by two cytokines, IL-12 and lymphotoxin alpha, to another module that is very loosely, much more loosely connected in chronic fatigue than it was in healthy controls. And this module captures the inflammatory and the Th17 response, which is a defense against fungal and bacterial infection. So again, probably one of the most striking features is interleukin-12 and lymphotoxin alpha. Interestingly enough, both of these cytokines are produced in large quantities by EBV-infected B cells. EBV-infected B cells will use lymphotoxin alpha as a growth factor, and they'll disrupt the function of IL-12 to produce virally induced dysfunction of natural killer cell response. And natural killer cells are vital defenders in viral response. So even though we did not know the exact trigger that caused chronic fatigue in these individuals, we found some interesting hallmark signatures of latent viral infection. So this only spurred us on in looking at a cohort, the Chicago CFIDS cohort, as I call it, where the trigger was now very well documented. It consists of a group of 300 adolescents recruited in the Chicago area, all of which were recruited upon diagnosis of infection with mononucleosis. And these individuals were then followed over the course of 24 months, and we can see that a number of them fit the case definition for chronic fatigue at 6 months. This fraction decreases at 12, and finally at 24 months we have 4% of the initial cohort that remain chronically fatigued 2 years after contracting infectious mononucleosis. So we were very interested in why this would be. It's important to note here as well that our control population is not a healthy control population. It is a recovering infectious population. So they're recovering normally from infectious mononucleosis. So one of the first questions that we asked was, well, these immune proteins, the same panel of immune proteins that we used to survey the adult Miami cohort, how are they co-expressed in this population of adolescents, and are the mechanisms that support their expression, the immune mechanisms that support their expression, are they shared between the post-infectious fatigue patients and the recovering controls? And the answer to that is yes. What we have here is the if the samples are in the lower left-hand quadrant of this graph, they essentially share the same patterns of co-expression. So the same mechanisms of response, of immune response, seem to be intact in the post-infectious fatigue and the recovering infectious mononucleosis patients. They're simply used in a different way and expressed to a different extent. So we were beginning to formulate the hypothesis that essentially a normal immune response might simply be misused in post-infectious fatigue. The second question that we asked ourselves, well, you know, this is fine. We have now established that these immune processes seem to be shared and they seem to be intact. Can the way in which they are expressed be used as a fingerprint to distinguish between recovering infectious mononucleosis patients and those patients who do not recover or may stay chronically fatigued? And the answer to that would be, well, as few as 5 of these 16 immune proteins that were surveyed, when taken together, when considered together, can separate quite nicely the post-infectious fatigue individuals from the controls. So the controls are the blue diamonds here, and the axis here is the degree to which we believe you are a post-infectious fatigue subject. So the controls have a low level of belief, of probability of being post-infectious fatigue, which they should, and the median probability for the post-infectious fatigue group is much higher. Interestingly enough, these 5 immune proteins also allow us to distinguish between 3 subsets of post-infectious fatigue subjects that were defined on the basis of the progression and severity of fatigue. So the first subset here, C1, represents a group for which fatigue dipped initially at 6 months but then increased rapidly at 12 and 24 months and stayed high. The second group represents a group where fatigue dipped initially at 6 months to a medium level and then plateaued out and stayed there, stayed offset from the controls. The third group represents, the C3, represents a group of PIPS subjects, or post-infectious fatigue subjects, for which fatigue initially increased but then decreased at 12 and continued to decrease at 24 months. So these C3 subjects, or cluster 3 subjects, represent what we might think is a more optimistic prognosis because they seem to be on a trajectory of slow recovery. And this signature essentially captures the elements of viral, allergic, and bacterial defense. In particular, we use interferon gamma, interleukin 5, and interleukin 23, and we also have some inflammatory metabolic markers, IL-6 and IL-8. So again, this is a rather narrow slice. It's 5 immune proteins, and we seem to be able to distinguish the post-infectious fatigue subjects rather well.
Gordon, could you speak up a little bit, please?
Of course, this brings us to the question of how generalizable is this fingerprint. Can we now look at adult CFS patients for which we do not know the trigger and apply this fingerprint? And the answer to that is in a minority of the cases, it would appear to be possible to do that, but the majority of subjects of 50-year-old adult female CFS sufferers did not demonstrate the same patterns of immune protein co-expression as the adolescent recovering adolescents and post-infectious fatigued adolescents. And whether or not the adults shared or did not share this pattern, this post-infectious fatigue pattern, was not related to age of the individual, to the age of the adult chronic fatigue sufferer, nor to her duration of illness. So this would be a separate or a different mechanism from what we believe we're seeing in post-infectious fatigue. So different, in fact, that the healthy adult controls are essentially more similar to, at least in terms of these 5 cytokines, more similar in immune signature to the post-infectious fatigue adolescents than the chronic fatigue adults. And so there's almost a reversal of polarity here. And in fact, when we drill down and look at why that might be, we have a reversal in the expression pattern of IL-5 and IL-8 such that the adult chronic fatigue individuals more closely resemble recovering infectious mononucleosis patients at 24 months, suggesting that they may have this low level of viral response that is just chronically active. Conversely, the post-infectious fatigue individuals would have an immune signature similar to a 50-year-old, which raises the issue of premature aging of the immune response. So as I mentioned, this is still very much an active project. We are currently waiting on genomic data from the Hussman Institute in Miami. So we will be looking at that very seriously. We have a student coming from Israel to work on this with us. This, as Kathy and Alan mentioned, this has been invaluable for us. The results that we have generated and the methods that we have developed with the CFIDS funding has allowed us to apply. We've used it as preliminary results in several applications, 3 of which were awarded us. We've been awarded, again, on the basis of our work with CFIDS, 2 U.S. Department of Defense grants, and very recently an NIH R01 for a total of $4.5 million in government funding that would not have been available to us had it not been for this seed money from CFIDS. It has also allowed us to enter into a very effective research network, and we've been collaborating quite actively with other CFIDS-funded teams, in particular the Climas Fletcher team in Miami and the Meadows Stewart team in New York. And so students have been going back and forth between my lab and theirs. In fact, this project alone links 5 institutions internationally. Finally, we very much believe that we should use an opportunity such as this to educate the next crop of practicing clinicians, and we have been doing that actively. CFIDS has been sponsoring a number of undergraduate medical students to do summer research studentships in my lab and introduce them to CFS research and how it is a complex condition and what we do know and what we are trying to understand about CFS, and then return them back to the pond and hope that when they become practicing clinicians, they will remember, they will have a different perspective on CFS as a condition and be better equipped to help this patient population. Finally, we've been communicating this our results as aggressively as possible. We have 1 paper published, 2 currently under review. We have given 2 peer-reviewed talks, and we have been invited to given a number of invited talks at several institutions based on our work with CFIDS. So again, it has been an amazing opportunity, and we'd just like to thank very sincerely the patient sponsors for their help and their trust. Thank you.
Thank you very much, Gordon. Absolutely fascinating talk, and it harkens me back to the days when I was in the lab doing all this work on genomics and the blood, and being quite worried that we might not necessarily be able to find some kind of signature in the blood that could help us understand what's going on in CFS patients, or serve as some type of signature, or provide clues or give us particular biomarkers. And I think what your work has shown and what the Light's work has shown is that those biomarkers are there in the blood. Now it's just a matter of obviously pursuing this work further and then getting reproduction and validation of these various markers so that we can really bring them into from the bench to the bedside. What our research support program is all about, bench to bedside translational research support. So our next talker is Dikoma Shangu. Dikoma is going to tell us about the brain and some of the research that he's been doing for about the past 4 years now. And again, some really fascinating work that's implicating not only biomarkers, but some of the pathophysiology that occurs in CFS. Thank you, Tacoma.
Thank you, Suzanne. And again, I would like to second what had been said about CFIDS Association's support of research. I really came to this, I could say, through the back door because I am a physicist, and I just kind of lend my expertise to anybody who's interested in conducting neuroimaging studies. And I was at Columbia in early 2000, I think it was around 2001, when I received a patient from Dr. Paul Cheney, and he just wanted us to do a metabolic workup on the patient. And at the time, we were actually studying mitochondrial dysfunction, people who had primary mitochondrial disorders due to some kind of mitochondrial DNA mutation. So when we did do a proton spectroscopy, you can think of it as MRI, except that MRI looks at the distribution of water in a tissue. Here, you're looking at MRI of the brain. And what proton spectroscopy does, it looks at everything except water. So we are looking at the spectrum here, which is why it's called spectroscopy, because you have a spectrum of peaks whose position along this x-axis determines what it is. So we can look at choline, which is a membrane constituent. We can look at creatine, which is part of the cell energetics in the brain. We could look at N-acetyl aspartate, which is a neuronal marker because it's a compound that's found almost exclusively within the neurons. And then we can look at lactic acid. And when we looked at Dr. Paul Cheney's patient, we saw a distinct lactic acid peak. Here, its frequency is well known. It's usually comes up as a doublet. So there was no doubt that this was lactic acid, also known as lactate. It was increased. If you look at, and the location of that increase was actually in the cerebrospinal fluid, which is contained within the ventricles, the lateral ventricles. And we're very puzzled by this. So, but we just kind of reported to Dr. Cheney that we thought that there might be a mitochondrial involvement because we are doing studies with patients who have primary mitochondrial disorders, and what we see is increase of lactic acid. And then he got very excited and he sent us another patient. We saw basically the same thing. And then over the next 2 years, we looked at 30 patients with a CFS, and at least half of them had this pattern. Became very intrigued, thinking that this is something that might be useful for understanding. At least it might become a signature for people with chronic fatigue syndrome. So we applied for funding from the CFIDS Association, and about 4 years ago we conducted the first study, which was to use this same approach using magnetic resonance spectroscopy, or MRS, to try to see what proportion of the in a control study, because the prior studies were just a really clinical workup and it was not controlled. We didn't compare the pattern that we are seeing against healthy control subjects. And also something that we wanted to do was to compare this pattern with another disease that often overlaps with chronic fatigue syndrome, and that was generalized anxiety disorder. And then we looked at, now we have conducted the study twice, and this is really has been the main observation. We have found out that the large proportion of people with chronic fatigue syndrome have this elevation of ventricular lactate. Compared to generalized anxiety disorder, we saw very little elevation. In the healthy volunteers, of course, you see very little of it. So, but then we did a second study where we compared lactate, lactic acid in the cerebrospinal fluid of people with depression. And there, although the value or the magnitude of the lactate was decreased, we didn't find it to be significantly different from chronic fatigue syndrome. And we think that probably if we can do this in a larger study, it's going to help us resolve that question. But the thrust of our research...
To point to the spot in the brain where you're actually finding the elevated lactate and describe that a little bit? Yes,
this is the cerebrospinal fluid. It's contained within lateral ventricle. It's just a space within the brain that this is a relatively low resolution MRI. But if you have a high resolution MRI, what you see is that this area in red is actually a fluid-filled structure known as ventricle or lateral ventricle. It's just a cerebrospinal fluid that's filled with it. So within this area, if you so the arrow shows that we are actually looking from the center of that structure, and that's where we are finding the lactic acid. We look within the brain, we don't see it, but there are reasons related to physics why we're not seeing that. We don't think that it's the lactate is actually being produced in the CSF over the ventricular space. We think that it's probably being produced in the brain tissue, but for physics reason, the intensity is too low to be detected. But it accumulates into the CSF, where it builds up in sufficiently high concentration to be detected. So this red structure is the area from which we are detecting the lactic acid. So yes.
Hey, Tacoma, you got about 7 more minutes.
Okay, so that we then became interested to try to understand why we were seeing lactate increase in people with chronic fatigue syndrome. I'm trying to move this so it's not up. Here we go. So we went to the literature and found the model proposed by Dr. Marty Paul, which implicates oxidative stress. And the observation that we had was that lactate was elevated in people with chronic fatigue syndrome. And the question is, why would it be elevated? Again, Dr. Paul's model of chronic fatigue syndrome, along with our own prior study of people with primary mitochondrial disorder, told us that when you have defective mitochondria, dysfunctional mitochondria, that will lead to impaired energy production in the form of ATP. But when ATP is low, another alternate mechanism for generating energy known as anaerobic glycolysis kicks in, and the end product of this mechanism is elevation of lactate. Another model approach that Dr. Paul had proposed was that you might have an infection, and an immune response to that infection generates free radicals. And free radicals, when they are not scavenged or buffered by an antioxidant like glutathione being the most abundant antioxidant in the body, then you're going to have a condition known as oxidative stress. Oxidative stress is when you have more free radical than the antioxidant capacity can actually remove the toxins from the system. Oxidative stress actually has been shown to lead to mitochondrial dysfunction. When you have dysfunctional mitochondria, they would actually generate more free oxygen radicals. And one of the mechanisms by which oxidative stress becomes deleterious to the cell is by attacking the phospholipid membranes, and through a number of reactions that leads to the formation of isoprostanes. releases isoprostanes in the circulation. It could be in the blood, it could be in the urine, it could be in the cerebrospinal fluid. So measurement of isoprostanes are a way to establish whether oxidative stress, which we have postulated, is actually playing a role in the pathophysiology of chronic fatigue syndrome. So you can see how we can begin to validate this model. We have the ability to measure glutathione. We have the ability to measure the isoprostanes in the blood. We have the ability to measure ATP. And of course, we have the ability to measure lactic acid. But isoprostanes, if when the increase in system actually lead to vasoconstriction, the blood vessels get constricted, and the consequences of that is that you have a reduction of blood flow. A reduction of blood flow almost always leads to increased anaerobic glycolysis because you don't have enough oxygen present, and of course the end product of anaerobic glycolysis is elevation of lactic acid. So this is a model that we are currently trying to validate, and the way we think we can validate it is again by measuring the level of isoprostanes in the blood of people with chronic fatigue syndrome, compare those values with those in people with depression and as control, and people in healthy control subjects. And it has been determined by a study by Kennedy and colleagues that isoprostanes are actually elevated in chronic fatigue syndrome. So we're trying to see if we can replicate that. We'd like to measure brain levels of glutathione, which is the most abundant antioxidant in the body. So the implication for this is that if we find that glutathione is normal and isoprostanes are increased, that tells you mechanistically that the antioxidant is actually overwhelming the defense system of the cell. Therefore, one treatment approach that one could take could be to try to increase the antioxidant capacity of cells. So what I'm showing here is the way we can use proton spectroscopy. Again, you're seeing this spectrum, a whole bunch of sticks, and we have the ability to cleanly isolate glutathione, and by doing some modeling, we can actually get the area under that peak, which is actually directly proportional to the brain concentration of glutathione. Another way we would validate this method is that since isoprostanes can increase or decrease cerebral blood flow, regional cerebral blood flow, we are using an MRI method to directly measure blood flow in the brain. And if blood flow is decreased, that might also explain why we might be seeing increased lactic acid in the brain. And finally, we would like to see whether there's a mitochondrial dysfunction. And the way we are doing that is using phosphorus spectroscopy and other magnetic resonance spectroscopy, which it's exactly the same technique as MRI, except here we are measuring compounds that contain phosphate and ATP, the cell's energy currency, and creatine phosphate, another. It's a storage for energy in the brain or in the cell. We're trying to measure these compounds in the living system to ensure that I'm sorry. I'm sorry. That was my telephone ringing, so I should have turned it off. So we're trying to measure these high energy phosphates, which are going to give us an idea of whether the mitochondria are functioning properly or not. Okay, so the outcome of measure here would be a direct measure. There's no technique currently that allows one to measure mitochondrial function directly in the human brain or in the human tissue. So if we determine that ATP or creatine phosphate are decreased, then that would implicate mitochondrial dysfunction in chronic fatigue syndrome. And because energy production is decreased, anaerobic glycolysis will kick in, and the end product of that is lactate. Thanks to CFIDS Association, we've now published 3 papers which basically report our lactate findings comparing with the depression and anxiety disorder. We have had the 7 presentations at international conferences. Most of them were actually accepted as oral presentations. These data, we can't wait to process all of these to try to compete for a larger finding, to try to validate the model. And again, our ability to give presentation at international conferences, we believe it's contributing to awareness of chronic fatigue syndrome, and we are really thankful to the CFIDS Association for the support. Thank you.
Thank you very much, Tacoma. It's another fascinating talk, and I think it's really providing some important clues to help us kind of get to the bottom of what's going on with CFS. I'd like to actually start with a question for you, Tacoma.
Yes.
What you show seems to be a very vicious cycle with this oxidative stress. You know, you connected these chemicals in the body and these organelles in the body with arrows, and they kept going back and forth. And it seemed like one, oxidative stress and mitochondrial dysfunction had some kind of almost crosstalk to it.
Yes.
When something is kind of a circuit like that, is it harder to maybe interrupt that circuitry without causing some other kind of effect, or in fact, is it easier to interrupt that vicious cycle?
I think it's probably more difficult because then you need to be able to find exactly where the reinforcement mechanism is. For example, you're absolutely right with respect to oxidative stress and mitochondrial dysfunction. It has been established in Parkinson's disease or Alzheimer's disease. These are self-reinforcing phenomena or events. So I think that it becomes very difficult because it's like there's a feedback loop that keeps amplifying the system. So if you can find exactly a point at which you can interrupt it, then you might. But the problem is that that's so interrelated that you can stop it at some point, but unless you actually can stop it at several points, probably you might not be able to stop it immediately on this track. Yeah.
That's right. Given that mitochondria plays such a big role in your model, do you think that there is possibly some genetic disorder in the mitochondria, or do you think it's some other kind of functional disorder rather than it's a genetic basis? With the mitochondria, I mean.
I don't think it's a genetic, because a lot of the work I did while I was at Columbia was actually looking at primary mitochondrial disorders that are actually genetic. And I think that, if I'm not mistaken, one of these patients we had studied at Columbia did undergo a complete genetic workup at Columbia. And there was no evidence of a mitochondrial dysfunction, a primary mitochondrial dysfunction. By that I mean that people who actually have in genetic makeup some signs of either mitochondrial deletion or mutation or rearrangement. And also if it's a genetic, then I think that it would tend to probably be familiar in the sense that, as you might know, we get all of the mitochondria from our mothers. Therefore, this is one of the hallmark of these primary mitochondrial disorders, is that if one member of the family has it, chances are that other members of the family will have it, because they would all gotten the genetic defect from the mother. So I don't think it's really a primary mitochondrial dysfunction. I think it's secondary, and the initiating insult, I think, is oxidative stress.
I think you're right, and your model nicely shows that this oxidative stress can in fact occur in the periphery, in other parts of our body other than the brain, and then have this effect on the brain. Elevated lactate, which obviously results in a number of, or contributes to a number of symptoms that people with CFS experience.
Yes.
One other question came from our listeners, Tacoma, and you described both general anxiety disorder and major depressive controls, and I think they wanted to know why you chose both those types of disease control groups.
There are two reasons. One of the reasons is that both depression and generalized anxiety disorder are prevalent among people who have a CFS. That was the primary consideration. A second consideration is that we just kind of looked at our expertise. We were working with Dr. Sanjay Matthew, who's actually still part of this study, who's done a lot of work with mitochondrial disorders, as well as with mood and anxiety disorders. Therefore, the fact that there was overlap between chronic fatigue syndrome and mood disorders, as well as we had expertise on our team, that made it the logical choice for us to use as a control.
Yeah, that's a great explanation. I think it's even more important to note that, in fact, when you measure these markers, they can differentiate people with CFS from general anxiety disorder and major depressive disorder. So very nice, very potentially important markers and processes that are going on in that you're learning about in this research that's really helping us understand what's going on with CFS.
I was just going to say that our drive is really to try to understand the mechanism by which some of the events we are observing might be contributing to chronic fatigue syndrome. So this is really a mechanistic study, and if you can actually understand the mechanism, then some of the parameters we are measuring might actually have the added value of being biomarkers. Until we actually understand what's going on, it's very difficult to use them as a biomarker for the disease, because they're not going to be specific enough, because we really don't understand what they are doing.
Right. And I think it's also important to note that what Gordon talked about and what Kathy and Alan talked about in their talks was with processes going on in the periphery, which in the blood, which actually can cause oxidative stress, or are likely causing oxidative stress, whether it be longer-term oxidative stress as a result of a persistent infection, for example, with EBV, or whether you get oxidative stress or some type of pro-inflammatory response, which is also a stressor in the periphery following exercise. So your work.
Absolutely, because I'm sorry, I was going to say that actually exercise itself has actually been established to be a big generation of oxygen radicals. So I think you're absolutely right about that.
Great. Okay, let me ask Gordon a question. We had a question from our audience on why were only girls in the post-infective fatigue cohort at 24 months, Gordon?
That is interesting, isn't it?
Yes, indeed.
Well, I think what experience I've had at looking at various data sets has indicated, at least from my perspective, that if I were to put a rough ratio to it, that chronic fatigue would affect at least twice as many women as it does men, and that it presents in a very different way in women than it does in men at the molecular level in particular. And that's not all that surprising. If we consider the hormonal differences between men and women, as the French would say, vive la différence. Again, it's not surprising that you would have a complex illness like CFS that involves the endocrine, nervous, and immune systems being quite different in its presentation between men and women. So what we, I guess what we're seeing with the Chicago cohort is essentially just supports that even further. We started off with a balanced group of young men and young women, and after 24 months we end up with a group composed exclusively of young adolescent women that are chronically fatigued after 24 months. So that 4% is entirely composed of young women. So you know, again, this is consistent with at least my observations of various data sets that I've been working with.
I really like, too, the way that the networks and this immune signature that you're finding implicates natural killer cell abnormalities or dysfunction. And, of course, we know that that was one of the first observations made by Nancy Klimas and Marianne Fletcher back in the early 90s. So it's again, I think fascinating with you're using a different set of tools to find, and I think in many ways validate an observation that was made very early on, and a very important observation that not only implicates immune dysfunction but possibly the cause or trigger of CFS, that being some type of viral activation or viral infection.
That's correct, yes.
So do any of our other speakers have a question for Gordon or Dacoma while we have him, before I go on to asking some questions to, why don't I just go on right before I open it up to everybody. Let me ask a few questions of Alan and Kathy. Sorry, that was a little spastic there. But Alan and Kathy, very important, cool work. And somebody in our audience asked, what's it going to take to get this test approved for clinical use? Don't forget to unmute. Kathy? Can anybody hear me?
Yeah.
I can hear you, Suzanne.
Can you hear
okay.
me?
I'm also enjoying Dacoma's music.
Yeah.
Hey, Alan and Kathy, are you there?
Yeah, we're here, but can we
Hear
hear
the
you?
question? What does it take to get tests like yours, a test like may be approved for clinical use?
Can you hear me?
Yes.
Okay, good. So I don't know if our speaker quit working or what happened there. Okay, so I'm on the regular handheld handset. Yeah, the problem is it depends on what you mean by clinical use. The difficulty is that the FDA has just passed, as you know, some new regulations in terms of diagnostic tools that make it a little more, at least a little more, although it's unclear how much more difficult it will be to actually get something FDA approved as a diagnostic tool. And that is as opposed to using it as a readout for treatment. So it's actually, you can do the readout for treatment much easier than the diagnostic test. Okay, so the real problem here that we run into, and it's sort of both one of those unexpected consequences, is that until we really understand whether there is a murine virus cause, like an XMR virus, actually causing chronic fatigue syndrome, nobody will be particularly interested in our biomarkers, because it could be all you need to look for is the virus. And this, of course, happened last October, you may remember. So as a result of that, we could not push forward and actually get the funding from startup companies to actually try to get a much wider dissemination of our test to try to find out whether it really can be clinically useful, that would actually be accessible to the FDA. So as a result of that, it's been put on hold until we figure out about the retrovirus story. And every week we keep hoping it will be solved. And we've been working on it ourselves since last October, and it hasn't been that easy. So what it would take then, so what it would take is, first of all, solve that question. And then second of all, a series of course, much larger ranging, much wider population of patients. to look and see whether we can achieve the same discrimination that we've been able to achieve with our cohort here in Salt Lake City.
I'm having a little bit of problem with that logic that the pharma companies are using, because it seems to me that you have a pretty well-established set of markers that have been validated and are not only able to detect certain subtypes, but are also able to be used as an outcome measure in response to certain types of treatments and as disability marker. So it seems like it's quite a diverse set of markers that can be used for a number of things.
Yeah, and they were truly interested, because we did approach them last year just about this time, and then of course the other thing came through. The problem is, as I said, that if all you need to do is track the virus, it's actually easier to track the virus than to actually look at our biomarkers. Much cheaper, because you're basically looking at a single PCR product as opposed to multiple ones the way we do it. So again, you can see the problem. You can spend a lot of money, and they don't want to put the money into the final establishment of the diagnostic biomarkers based on our system until the virus question is solved. Does that make sense?
Yes, it does. It does.
Yeah.
I still think there will be plenty of room to use your biomarkers when we know more, even a little bit more about XRP, but not to belabor that point.
Yeah, in fact, we think that as well, and that we've actually, and one of our attempts here has been to try to relate any viral infections to the our biomarkers. So yes.
Great. So then it sounds like maybe it would be interesting also to look at your biomarkers in cohorts such as the one that Gordon's studying, the post-infected cohort.
Yes, very much so.
That's one of the reasons, one of the things that we're doing with our biobank is to create a resource where investigators like all of you can turn to a centralized resource in order to be able to further validate your markers on other populations.
Yeah, we are working with Lynn Jason to try to do at least some of that in essentially the same cohort as Gordon is working on there in Chicago. So yes.
So great, you have validation studies going on already.
Right.
Super. All right, why don't we take the next 3 minutes or so to open it up to our panel, and if you guys have any questions for each other or for me, we can entertain a few over the next minute or couple of minutes.
Yeah, this is the comment. I think I was very intrigued by the results by Kathy and Alan. Have you looked closly at how well you're able to differentiate fibromyalgia from chronic fatigue based on your biomarkers?
Can you hear me, Dacoma?
Yes, I can hear you.
Okay, good, because we were having trouble with our speakerphone before. Yes, we have actually, and that's we actually see substantial differences between patients who have fibromyalgia only versus patients who have chronic fatigue with fibromyalgia and then chronic fatigue only. And that data, unfortunately, is being held back right now because we haven't been able to publish it. We have not put it out for publication anywhere yet, so it's nowhere right now. But we do see substantial differences between them, and it's really interesting, very, very interesting in terms of what happens with the fibromyalgia patients. I'm sorry I can't tell you more right now. I'd like to.
Yeah, I think that'll be fascinating, because I think that's really one way to get at the specificity of your biomarker, considering the similarity, or at least the overlap between chronic fatigue and fibromyalgia. So I found that to be rather fascinating.
Oh, so did we. As you will see in the very near future.
Can't wait.
So far, all of the tests that we've done comparing to other groups, we have the signature profile of the chronic fatigue patients is unique. We have not yet found any other patient population that shows this pattern. That's so far. Now, as I say, one of our goals is to look at many other patients who experience fatigue as part of their symptom array, but so far no other group has this signature profile.
Have you looked at depression?
Yes. Yes, we've looked at major depression. We actually are doing a totally separate study looking at gene expression in depression. And no, they don't have the same profile either. So.
More fascinating.
Yes, outstanding.
They have an interesting profile of their own, by the way, as you have already indicated. But
yes.
Yeah.
This is Kim. I'm going to jump in and ask a question that came from the audience. Why is it important for you guys to have pilot data when you go to the NIH for funding for larger amounts than we're able to provide?
Okay, who wants to answer that one? I can give you our it goes this way. NIH is way underfunded. We'll start right there. If you look at the amount of money that's put into pain research, for example, it doesn't even approximate the amount that Pfizer spends on commercials on their pain products, or even one product. Okay, so there you go. There's very little money at NIH to actually support any research. Okay, if you're looking at a whole series of grants, and we do this. I'm on the review committees, and I know a number of rest of you are as well. If you have 5 grants, and one of them has pilot data showing it's very likely to work, and you have 4 grants that don't have any pilot data, which ones are you going to fund? Which ones, and you have no idea because there's no pilot data whether it's going to work at all. Well, you're going to fund the one that has the pilot data. Does that answer the question?
Yeah, and I think it's also important to point out that there are different categories. So there's a category of funding that will take ideas without preliminary evidence, but still the grants get put in this particular category with preliminary evidence, and they're just more attractive to reviewers and to funding agencies because it's a lot less risk.
That's it.
I think that's the key. I've had a grant, application funded by NIH, and they really got funded when I have absolutely shown them that I can do it. That means I've done so many preliminary data that essentially I'm asking them to fund a study that I have already done. So you just kind of use the money to actually take a new direction or just kind of enlarge the study. So I think that it's the question of risk because the funds are so limited.
Okay.
Gordon, do you have anything to add to that?
No, I couldn't agree more. I think it is a risk reduction exercise, and I come from a private sector background, so we were all about risk assessment, and we would always go through these stages of pilot and staging and then full scale.
Yeah.
So it's quite consistent with my engineering background.
And also something that's important, Suzanne, you just mentioned that there are mechanisms that you really don't need preliminary data. The problem with that is that the funding for those mechanisms are really limited. Essentially funding you to demonstrate a unique concept so that you can get preliminary data and apply for the R1, which is actually the mechanism that funds most large studies.
Yeah, so I think the funding that the CFIDS Association is able to provide is incredibly important to investigators like you've heard from today and to the rest of our funded investigators. And we hope to be able to have a successful rest of the year in fundraising to continue this incredibly important program of supporting research such as this. So we hope, if we're successful, if you like what you've heard today and you want to hear more, please help us in reaching our goal of $5 million by the end of the year. And we'll be able to support this kind of research and build on the research that you've heard today in order to be able to get markers from the bench to the bedside to impact and improve the lives of people that suffer with CFS. The slide that's up now has a number of URLs available that you can go to, and also don't forget to click on that Donate Now button when you go to these sites. And we can also we will be posting this webinar if you want to hear it again, or if you know somebody who missed it and wants to hear it. It'll be posted also on our www.cfids.org site. All of our webinars are actually posted. It'll take about a day or two to get this recording up. And yeah, you can follow us on Facebook and all of our other nice URLs that are listed on there. But please, if you enjoyed today and you believe that research is the way forward, help us do what you can in order to help us raise that money to continue to support this kind of research. We are the CFIDS Association with a very, very important mission of diagnosing, well, making CFS widely understood, diagnosable, curable, and preventable. And we do that by funding research like you heard today. So thank you very much we had a great audience. Everybody stayed with us the majority of the time. We've had some good questions come from the audience, which we were able to get in there, and some great presentations. Thank you, all of our investigators, all of our presenters today. That was fabulous. You all stayed on time. It was fun. I wish we could listen to each of you for about 2 hours to get down all the details, and hopefully we'll be able to do that as we continue this webinar series. Thank you again, everybody, and have a great afternoon.
Thank you.
Thank you.